Okay, so good afternoon, everybody. I'm sorry that we ended up having to do this class remotely, but needs must as the devil rides, so they say. This is actually one of my favorite classes to teach. That's why I'm kind of a little bit miffed. Anyway, so we're going to start talking today about the invertebrates. This is a very complex topic. I, in fact, have taught this topic over an entire semester, and I myself studied it for 2 semesters for an entire year in invertebrate zoology. So compressing it into one class is quite an undertaking. So, but let's start off basic definitions. Of course, invertebrates are animals which lack a backbone. Now, that backbone does not have to be actually made of bone itself. There are vertebrates who have a backbone which is made of cartilage. But the invertebrates are those which entirely lack a backbone. And they account for a huge proportion of the animal kingdom. 95% more of the animal kingdom are actually invertebrates. First of all, an animal, what is an animal? Animal is multicellular heterotroph. And the key to that definition of what an animal is the issue of multicellularity. Multicellularity implies that the animal has a collection of cells which coordinate with one another to construct an entire organism. And we believe that the animals all arose from a single common ancestor, as we heard previously. And that single common ancestor was a choanoflagellate, a single-celled protist, choanoflagellate, which we discussed before. We see the same cell type in a particular group of animals, which most biologists would agree were true animals, and those are the porifera, the sponges, where choanocytes, actual cells very, very similar to choanoflagellates, are a functional part of the organism. Okay, so here is a basic phylogenetic tree for you. And we are going to be discussing all of these groups, noting that the vast majority here, all of these groups here only include invertebrates. But when we come down here to this clade here called the deuterostomia, the deuterostomes contain both invertebrates and vertebrates. In fact, we are in this group. We are in the deuterostomia, but we are kissing cousins to a number of invertebrates, as we'll hear later on. Okay, so first of all, let me just point something out about this tree. That is, it has a basal group there. That is the basal group, the porifera, the sponges. And in fact, there are some biologists who would argue that the sponges are not true animals because their cells are not organized into tissues. So that we place them outside as a basal group, and all of the rest of the invertebrates, all of the rest of the animals, in actual fact, are clustered in a clade called the Eumetazoa, the true metazoans. That is those organisms where cells have actually organized to form true tissues and then true organs as well. The porifera don't have that. The porifera, a very, very ancient group. We see roots of the porifera way back before the Cambrian, in actual fact. And they occur, they're aqueous species. They occur predominantly in marine systems, but a few in freshwater as well. Fairly loosely organized. Their cells do not form true tissues, but the cells have specialization of function. And these, and some sponges in fact, are quite well organized and quite highly specialized. The most important thing to note is the importance of this cell here, called the choanocyte, which you'll see has very, very strong similarities to the choanoflagellate. And those similarities, by the way, extend to molecular similarities as well. So it's quite clear that the choanocytes and the choanoflagellates are closely related. Here's a very simple sponge, which is organized as follows. It has a wall around the outside. And then in the center here, there is an opening. And this wall is perforated by pores. The choanocytes beat their flagella in order to create a current which passes through the pores and then out through an opening called the osculum. Now this, in many sponges, this simple form gets elaborated on. This is just one simple sponge. But the point is that in the pulling water through here, what the sponge is doing is it's pulling in oxygen, obviously, to oxygenate the tissues, but it's also pulling in material which could potentially be food, which is filtered out by the choanocytes. Throughout the wall of the sponge here, there are different kinds of tissues, and one type of tissue, one type of cell rather, I'm sorry, don't use the word tissue. One type of cell is an amoeba site. The amoeba sites actually feed on material plucked out of the water by the choanocytes and distribute it throughout. There is no nervous system, but in fact, the sponges can respond to stimuli. If you poke them, parts of the sponge may contract. There are cells which are able to contract, precursors to muscle cells, you might think, which are also sensory, but there is no true nervous system. Very simple, very simply organized clusters of cells, some of which have specialization, but not true tissues. The next group that we're going to talk about are these, the Nidaria, and these truly belong in the eumetazoa because their cells are organized into true tissues. But in actual fact, they only have two layers, two cell layers, two layers of tissue, one on the outside called the epidermis, and then one on the inside called the gastrodermis. Could be called the endodermis as well, but you can call it the gastrodermis because that will remind you that this is actually forms the gut. So the basic form is a bag, right? And it has one opening. It has one opening which functions both as mouth and anus, lined with a true tissue, and then an outside lined with true tissue. And there are two body forms in this group, the Nidaria. The first is called a polyp, and that is a tube usually fixed to the substrate. They can move very often. Things like sea anemones can move slowly, but other times the polyps are colonial and form things like the corals. But their basic form is this, a tube with a single opening going into a gastric chamber where all the digestion and everything takes place here. All the waste has to go out the same opening. The other body form in the Nidaria is that of a Medusa. And this is probably familiar to you as the jelly, improperly called the jellyfish. It's not a fish at all. And properly, it should just be referred to as a jelly. And look, and you'll see the similarity in the body form between the polyp and the Medusa. And the Medusa is really like an upside down polyp that is free in the water. It is suspended in the water column and is motile, usually by pulsing water in and out of the bell underneath here. So those are the two of the basic body forms. Many of the Nidaria alternate in their life cycle between one and the other. So, for example, the big jellies that you are probably familiar with, they have a polyp stage earlier on. And then some of the others do not ever have a polyp stage, both stages. The sea anemones, for example, never have a Medusa. That's many of the Nidaria alternate between one and the other. The one thing which unites all of the Nidaria together is possession of stinging cells. And the stinging cells are present predominantly on the tentacles, but sometimes on the body as well. And the stinging cells have two purposes. Let me just go here. The stinging cells have two purposes. One is for predation. The other obviously is for protection. And they may contain extremely powerful toxins. So this is what the cell looks like. It is called a Nidosite or nematocyst. You can use either word, I don't mind. There's some distinction between the two, but it doesn't much matter. You can see here this is the nematocyst is coiled up inside out, inside the cell here, and it's kept under pressure. And there is a trigger. If you touch the trigger, it discharges very, very powerfully. And it can actually embed itself into the tissue. And there is a sac of toxin which is discharged. And those toxins can be extremely powerful. Very, very often, they will actually start to break down cells. They are enzymatic, and they'll start to break down the cells that they come in contact with. So both for predation and for protection. The Nidaria, a very, very ancient group, way before they originated, way before the Cambrian. They're extremely diverse. Here are just a couple, and never mind of much about the Latin names. But this is a jelly, typical jelly, with a bell, which it uses for locomotion, and which contains the gut, the simple gut, and the reproductive system. Very importantly, the Nidaria have a complete nervous system, but it is like a net. They do not have a brain because they are radially symmetrical. So they do not have any cephalization. Their head doesn't develop at the front because they have no front. Instead, the nervous system is like a net throughout the whole organism. Below here are all of the tentacles and everything covered with their stinging cells. Now, those are the jellies. Remember that these jellies have a polyp stage. Here's another of the Nidaria. This is the Portuguese Man of War, well known because its toxin is extremely powerful and causes actual almost burning of the tissue that it comes in contact with. Here is ordinary sea anemones, very, very common, and that's the typical polyp form, the tube up the top, the tentacles with the stinging cells. And in between here would be the mouth, what would be the mouth anus, because it's only a single tube, a single opening leading into the stomach here. This is called a soft coral. These are the hard corals here. That's hard coral there. Here's another soft coral there. Now, the Nidaria, there are some of the polyp forms of the Nidaria that have a very small Medusa stage, and this is usually the sexual stage. In fact, you see these 4 dots here, 1, 2, 3, 4. Those are the gonads. And this would be no more than 1 millimeter or 2 across. And it's relatively short-lived, while the polyp is long-lived. In the jellies, the polyp is short-lived, and the jelly itself is the long-lived form. Okay, so those are the Nidaria. The first of our the true eumetazoan groups, and they're in the eumetazoa because they have true tissues, only 2 layers though. In between those 2 layers, there is what constitutes the jelly, so which we would call the Mesoglia. The other groups, the most important thing to note about them is that they're all based on a body plan which is bilaterally symmetrical. And so they are clustered in a clade called the Bilateria. And there are the following groups. You have heard these names before, but it's time to start learning the group names. First of all, the Lofo Trochozoa, Ecdysozoa, and the Deuterostomia. Those are the groups that we're going to be talking about. Remembering that these are all invertebrate. The deuterostomes divided between invertebrate and vertebrate species. Bilateria have the characteristic, they're bilaterally symmetrical, but not only that, they all show a tendency towards cephalization because they're bilaterally symmetrical and they tend to move in one direction through the environment. The front end becomes cephalized. It develops the predominance of the sensory organs, etc. Those may become extremely elaborate. And in order to service them and process the information, the nervous system begins to develop a centralized area for processing all of that and getting the whole organism to respond. The beginnings of a central nervous system begin to arise in the bilateral organisms. So our very first group that we're going to talk about are the Lofo Trochozoa. The most abundant of groups. There are lots of groups in the Lofo Trochozoa, some of them very obscure. The most abundant groups are the mollusks and the Anilids, and we'll discuss a little bit about each of those. So the first group that I want to talk about in the Lofo Trochozoa are somewhat simpler than the mollusks and the Anilids. And this is a group called the Platy Helmets. Platy Helmet literally means flat worm. And in common parlance, these are always called the flatworms. Again, another very ancient group, but they have the characteristic that they are triploblastic. They have 3 layers of tissue, where the Nidaria only had 2. From now on, all the groups that we talk about are going to be based on a 3-layered bilaterally symmetrical model. But the platyhelminths are still a primitive group because they have an incomplete gut. They still have a gut which is a blind-ended tube. So they have a mouth / anus. They eat and they pass waste through the same opening. They occur in a wide range of habitats, as long as it's wet. They occur in marine, freshwater, and in damp terrestrial habitats. You'll even find them in greenhouses. In tropical areas where you have damp forest floor, you may find them on the forest floor. And they are always flattened dorsoventrally. And that's where they get their name from, Platy Helmet, and flattened from top to bottom. They are cephalized, and they have a much more complex nervous system than the simple nerve net of the Nidaria. In fact, they even at the front already have little ganglia, which there's definitely not a brain, but certainly an enlargement and an elaboration of the nerve ganglia up at the front. Very, very complex reproductive tract, and often a very complex reproductive cycle and reproductive mechanisms. They also even have primitive kidneys and some other organ systems. So they appear to be much more highly organized than the Nidaria. So just by the way, they occur in many different forms. Many of the marine flatworms are extremely beautiful and very, 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 very striking in their coloring. This is one of them here. Some very large as well. A number of the flatworms, there are lots of free-living flatworms, but a number of the flatworms are also parasitic. And if in human and in animal terms, there are two particularly important groups. One are the liver flukes, as you see here, which there are a number of them, and they cause a wide range of different disease states. That's a liver fluke there. And this here is a tapeworm. And the tapeworms are also members of the Platy Helmets, but they are much simplified. They still have a nervous system, elaborate reproductive structures, but they are very much reduced because they live in a parasitic environment. And the tapeworms never have a stage which is actually in the open. They always acquired from one host to the other, although they may have different hosts. The liver flukes often have stages which are aquatic before they become parasitic. Here's a tapeworm. The tapeworms fix themselves into the gut with a head there with teeth on it, which embeds itself in the gut wall. The rest of the worm trails behind in the intestinal cavity. And it acquires most of its nutrition just by the diffusion of nutrients into the body of the worm. They have no gut, the tapeworms. This is a 59-foot long tapeworm recovered from one patient. They can reach enormous lengths. So they're quite spectacular organisms. Okay, so that's our first Lofo Trochozoan group, the Platy Helmets. And the really significant thing about the Platy Helmets is they are triploblastic, but they are solid. They don't have a body cavity apart from the gut cavity. All of the rest of the groups that we're going to talk about actually have a body cavity apart from the gut cavity. And the first group we're going to talk about are the Malaska. And the mollusks are very, very diverse. We see them most prominently coming to the fore during the Cambrian period, when there was the Cambrian explosion. Many, many, many mollusk forms arose. And for millions of years, there was a hugely important and very dominant marine group called the Ammonites, which persisted for hundreds of millions of years, right up until the same time as the dinosaurs disappeared. Now, most mollusks and the most molluscan diversity occurs in marine systems, but there are some freshwater mollusks as well, as you probably know. And we, of course, on land have slugs and snails, which are also mollusks. So they are soft-bodied animals, but many of them protect themselves with a hard shell. And that shell can take various forms, which gives us various different groups of mollusks. The most primitive of the mollusks are the chitins, and their shell is divided into plates. You can see there 1, 2, 3, 4, 5, 6, 7, 8 plates there. And those plates, if you prise a chitin off the rocks, which is actually very difficult to do, they will curl themselves into a ball with the plates protecting them on the outside. They are regarded as being a primitive molluscan group. The more advanced mollusks are the gastropods. And the typical gastropods are the land snails, for example, but all of the marine snails, the conchs, all of those things with a single spiral shell are the shelled gastropods. But there's also groups of gastropods which have lost the shell. They have secondarily lost the shell. And the most spectacular marine group are called the Nudibranchs. Now, if you want to treat yourself, and just go to Google image and Google this word nudibranch, or else go to Instagram and Google nudibranch. Just look up nudibranch and have a look at people's spectacular pictures of different nudibranchs that are a marine group that are shellless, slug-like gastropods. The slugs on land that we get on land are also gastropods in the same way. They have lost their spiral shell. One thing to note about not the chitins, which don't have them, but in many of the mollusks, they have eyes which are very similarly constructed to our own. But this is an instance of convergent evolution. The origin of Molluscan eye and the vertebrate eye are completely different, but their functioning is somewhat similar because the Molluscan eye also has a retina, and it may also have a lens. It doesn't always, but it may also have a lens, and usually has an iris, which can open and shut in response to light. So very similar in structure to the vertebrate eye. There are a large group of mollusks which have two shells, and these are called the bivalves, and the most familiar ones to you, probably the scallops and the mussels. We love to eat bivalves, very delicious, and for many millions of years they have served, mollusks in general have served as a major food source for human beings. And there are piles and piles of mollusk shells, which are hundreds of thousands of years old, left behind by our ancestors who scavenged seashores and fed on mollusks. So we know that we have a very long record of interaction with these. These are the scallops. Scallops are actually motile, and they swim by clapping the shells together like that. They also have eyes. You can see there these little dots there all the way around. Those are eyes modeled as I told you. There's an eyeball, the retina, and they actually have a little lens and everything. So on the inside of the bivalves here are elaborate gills. And the gills serve two functions. The first, obviously, is to oxygenate the animal. The second is for feeding. They actually filter food out of the water column using the gills as a sieve. Same thing here. These are the mussels. And this here is a giant clam, also a bivalve. This is from a tropical area, but exactly the same kind of model and lifestyle and everything else. These here are cephalopods. This is another molluscan group. Very, very extraordinary group. I hope you've all seen my octopus teacher. If not, look it up because you can get it on streaming. The cephalopods, a remarkable group, very, very ancient, dating all the way back to the Cambrian. And for many millions of years, a very dominant group in marine systems. We still have many different cephalopods. Cephalopod means head foot. And this is what has happened with the cephalopods. They have a large, very well-developed head with quite a large and elaborate brain. And at the front, they have tentacles of some sort or other. And usually a very large, well-developed eye, very similar in function to our own. So these are the squid, they have the long body. These are octopus, which have a short, squat body and long. Of course, you probably know that the cephalopods are remarkable amongst invertebrates in their ability to learn and apparently to do simple reasoning as well. You can present octopus with a problem, for example. You can present them with a jar with a crab inside and a lid on the top, and they will learn to unscrew the lid to get at the crab. Have I got time? Yes, I have. I used to work in an aquarium, and we had an octopus that we realized was climbing out of its tank at night. They have an absolutely uncanny ability to creep through the tiniest holes and slits. And this octopus was getting out at night. It was going along the walkways, which visitors to the aquarium can't see, which run above the tanks. It was going along a U-shaped walkway and going and catching fish in the tank that faced it across the gap, and then finding its way all the way back to its own tank at night. And we, for ages, couldn't work out why these fish were disappearing from the tank opposite. It was this octopus had sat there and watched the fish up in the tank opposite, and eventually worked out, okay, if I get out and I go around, I can go and help myself. So they really are remarkable animals. This is a chambered nautilus, the only living remnant of this huge group of shelled cephalopods called the Nautilus and Ammonites, which remain dominant in marine systems for hundreds of millions of years until around about 66 million years ago, when most of them went extinct, except for this one remnant group who live in deep waters. Well, they spend their day in deep waters in tropical systems, or usually off tropical reefs. And these are the chambered nautilus. One of the more primitive of the cephalopods. And one of the interesting things is, look at its eye. You see that it's got an iris with a slit in it. And this, unlike most of the other mollusks in the nautilus, the eye is actually open to the water. So the retina at the back is bathed with seawater. There is no lens. Instead, they form an image by contracting this pupil in here, this like slit, which they use to make an image on the retina at the back. This is the more primitive condition in the cephalopods. Okay, so that the mollusks are one of the very large diverse groups in the Lofo Trochozoa. And the other group in the Lofo Trochozoa are the Anilids. The Anilids are worm-like creatures, and they have bodies which characteristically have a series of repeating segments, often a very large number of repeating segments. And each repeating segment has usually the similar organ systems. So each, in an earthworm, for example, many of the segments, each of them has a primitive kidney in it. Each of them has a nerve ganglion, etc., etc., etc. Both the mollusks and the annelids are coelomate, unlike the flatworms. Both annelids and mollusks are coelomate. However, in the mollusks, the coelom has become very much reduced, and instead they have a pseudocoel or a hemocoel. You can call it either. It's either pseudocoel or a hemocoel. Let's go back to our previous class and look up what that meant. But the annelids are true coelomates. And the coelom in the annelids has various functions, but one of its most important functions is fluid filled. So it's used to conduct oxygen and nutrients and everything back and forth. It's filled with a blood-like substance. But quite apart from that, the fluid in the coelom cannot be compressed. Fluid is very difficult to compress. So if muscles contract around the coelom and then the animal will extend, and that gives the worm the basis for locomotion. So the coelom in this instance is acting as a fluid-filled skeleton, a hydroskeleton against which the muscles can operate. Similarly, if you take a portion of the worm, close it off, and imagine that its longitudinal muscles contract, then the worm will swell. Contract the circular muscles, and the worm will extend. This is the basis for its locomotion. Now, the group are called the Anilids, and they are divided into three dominant groups: the polychaeta, which are marine; the oligochaetes, which are the earthworms and all of their relatives, who get them in soil and in aquatic habitats, both marine and freshwater. And then this other group called the Hierodinea, and these are the leeches. Some systematists would put them together with the oligochaetes, others separate them out. And the leeches, most famous for being parasitic, but not all of them are parasitic. So we'll just have a look. The marine annelids, very important marine group and extremely diverse. This is the one form that you see very, very commonly. You can see all the repeating segments down there like that. And on each of the segments, there are specialized locomotory organs called the parapodia. And these have actual hairs in them, stiff hairs, which can be put into the substrate and used to move through the substrate. That's where they get their name from, the polychaetes. In other cases, these parapodia are modified to allow the worm to actually swim. These here are called fan worms, and they are sessile. They are embedded in the sediment, and their heads extend out and have these crowns on them, which are used for oxygenation, but also for feeding. They are filter feeders. They pull water current through these and filter out food particles. Here's the group that's probably most familiar to you. And these are the earthworms. This is a giant earthworm from Australia. But the earthworms are probably quite familiar to you. They differ from the polychaetes and from the leeches, the Hierodinea, in that they are almost always circular. The others are somewhat flattened. Leeches often very flattened, even though they have repeating segments. This group, the oligochaetes, are almost always circular in cross-section. I should just mention one interesting thing for you. Do you know, if you go into the garden or you go in Prospect Park or something, you just dig a little way into the soil, you lift up a rotten log or something like that, you will see earthworms all over the place. They're very common here. And we have some really large earthworms as well that occur in the garden. In actual fact, in our area, in the New York area, in fact, the whole of the northeast of the United States and most of the north of the United States and Canada, there are, in fact, no indigenous earthworms. And the reason for that is that the whole area, which was covered with ice by glacier during the ice ages, lost all of its earthworms. And all of the earthworms which we see in our gardens now have actually been imported, mostly in nursery stock and nursery soil, and in the topsoils that people buy at Lowe's and put on their gardens. Almost all of our, in fact, all our earthworms in the New York area are completely foreign, completely exotic to this area. Okay, so those are the Lofo Trochozoa. Remember the mollusks and then the annelids. We're going to talk now about this group here, which are the ecdysozoa. Now, the other thing I should mention to you is that this whole group here, plus some others, which are not shown here, are protostomes. But this is not, this group is not called the Protostomia. But it's important to remember that the Lofo Trochozoa and the Ecdysozoa are both protostomes, not deuterostomes. Go back to our class last time and see exactly what that means, exactly what their embryonic development and everything is. If I should ask you, if I take an earthworm embryo of 8 cells and I separate the 8 cells, what would be the fate of each of those 8 cells? And you should be able to tell me, knowing that they are protostomes, not deuterostomes. So the other group here in the protostomes are the Ecdysozoa. Ecdysozoan means animal that sheds its skin. Ecdysis is the act of shedding an outer skeleton, shedding the outer skin. That should clue you immediately. That should give you the immediate clue that what we're talking about are those organisms with an exoskeleton. And that exoskeleton is fundamentally called a cuticle. But that exoskeleton is tough, and it is hard, and it does not expand. So as an animal grows, it has to shed that exoskeleton and have some way of creating a new one. That's the first thing. The second thing is that exoskeleton is very hard, and so it has to be segmented so that the animal can locomote and move. And this is a characteristic of one huge part of the Ecdysozoa, but not of the other. The 2 phyla that are most important, there are some other minor groups that we're not going to talk about. The most important groups are the nematodes and the, excuse me, and the arthropods. And here is a nematode. It may seem very strange to have these beasts put into the Ecdysozoa. But the fact of the matter is that they have an outer exoskeleton, a cuticle made of this substance chitin. And chitin is a polysaccharide, a very tough polysaccharide. In the case of the nematodes, the exoskeleton is very thin and much more flexible than it is in the other group that we're going to talk about, which are the arthropods. Nonetheless, the nematodes have to shed this outer skin as they grow, and have to have a system to regrow a new exoskeleton afterwards. Nematodes are probably one of the most numerate of all of the animal groups. There are probably more nematodes than there are any other animal. And they occur in virtually every single habitat on the planet except the most extreme, like this middle of Antarctica or whatever. And I wouldn't bet that there aren't some there. They vary from being very, very small, microscopic, to being extremely large. Some of the nematodes may be feet long. They occur in a huge range of habitats and marine freshwater soils, and there are many, many which are parasitic on both plants and on animals. And some of our commonest parasites are in fact nematodes, because these are the roundworms. Almost every single human being at one point or another in their life, in fact, almost every animal at some point in their life, carries a certain number of these nematodes. And in children, there are 2 particular ones which are important. The first is called a pinworm, and the other is the common roundworm. And these are both intestinal worms. And very interestingly, the possession of a certain population of nematodes at some point in development appears to be very important in the development of a mature and fully functioning immune system. And in areas where nematode infestation is almost completely eliminated, you see a concomitant increase in the number of people who suffer from allergies and from autoimmune disease. We have evolved together with our nematodes over a very, very, very long period of time. And for the most part, carrying a certain number of nematodes is not harmful and may in fact be beneficial. It's a very strange thing to think about, but it is true. Carrying a heavy burden is not beneficial. There are some nematodes which do definitely cause real illness, but carrying a certain number appears to be completely acceptable on an organismic level. So the other group of ecdysozoans are the arthropods. And the arthropods are just so incredibly successful and so diverse that they could really be regarded as being one of the most successful of all of the animal groups. 2 out of every 3 known species of animal are arthropods, and we only know about a fraction of the number of different arthropods that there are. This applies in particular to 2 groups. The first are the crustaceans. The second are the insects. And those 2 groups, the crustacea and the insects, are hugely diverse and occur in many, many, many different kinds of habitat all around the planet. So this here is a relatively primitive arthropod from the Cambrian. This is a trilobite. And the trilobites, typical arthropod in that they have this tough exoskeleton. And the exoskeleton is made of chitin, of thick chitin, but very often with calcium carbonate incorporated into it. So it's rigid, tough, and hard. That is for protection. Particularly in these early marine species, it was for protection against the early animals that had jaws that were actively hunting. But her possession of that rigid exoskeleton means that this puts constraints on the animal itself. Because if you've got this rigid exoskeleton, how are you going to move? The only successful way to do it is to segment that exoskeleton, as you can see here. And in the primitive arthropods, in fact, this segmentation is much the same as it was in the Anilids, in that the segments tend to repeat their organ systems and everything. If we come to something, have a look here. This is a centipede. You'll see what I mean. The centipede has repeating segments. It definitely has a tough exoskeleton, and its limbs have to be jointed, as you can see on the legs here, in order for them to work. The exoskeleton, that word exoskeleton is absolutely accurate, because this is a true skeleton, in that the muscles are attached to the exoskeleton, and the muscles operate the exoskeleton. So the muscles operate against the exoskeleton, so that they move the body and they move the limbs. That's easiest to think about in something like the centipede, which is regarded as a primitive arthropod. You can still see segmentation even in the more advanced arthropods. These are arachnids. This is a scorpion here. And you can also see it in the crustacean. which are then in themselves an extremely large, very, very diverse group. So here's the Arthropod Origins lie back in the Precambrian, and it's the basic body plan is this exoskeleton jointed with repeating segments, and with the primitive condition was to have many, many limbs, many, many jointed limbs. There's little in the primitive ones, there's very little variation. Very often in more primitive arthropods, the head and thorax are joined together into a cephalothorax. So we have cephalothorax here, and then the abdomen. This is the fossil of a trilobite, and you can see that it has large compound eyes there. Were we able to dissect it, we would find that there's actually a fairly large collection of ganglia, a primitive brain here, and then underneath a double nerve cord running the length of the ventral surface, not the dorsal as in us, but the ventral surface. That's the basic body plan. Now, everything that we see afterwards in the arthropods is an elaboration of that. Let me go back here. The more primitive show the similarity of segments throughout the whole body. The more advanced arthropods begin as we move through the arthropods and watch development. We see here in the scorpion, the spiders, things like that, there is a cephalothorax. Here are repeating segments, but we're beginning to see that there is reduction of the number of limbs. In the arachnids to 8, in the insects to 6. The crustacea are a more primitive group. They have far more limbs. So if we look at the crustacean condition, what we see here is cephalothorax there with limbs attached. They still have quite a number of limbs because they have limbs which you can't see in this diagram. Here is the abdomen here, and the crustacea have limbs attached to each of these segments here. So this is a primitive condition. In fact, these swimming appendages here are modified limbs as well. The limbs, however, are becoming specialized. They're becoming specialized for particular purposes. The nippers, obviously for protection and for feeding, for predation. These are locomotory here underneath. The abdominal limbs here are also locomotory because this is often how they, one of the ways in which they swim. They can swim by just beating these abdominal appendages. And then these here at the end, the tail, are also modified for locomotion, but these act to, when the abdomen is flexed, to move the animal backwards. So the abdominal limbs for swimming forwards, and the flexing the abdomen rapidly pulls the animal rapidly backwards, usually in an evasion. They have very well developed eyes and quite a large brain as well. And then, as I told you, a ventral double nerve cord that runs all the way down. Yeah. Most of the crustacea have a larval phase, which looks completely different to the adult. And it's very often in aquatic systems, is very often planktonic, where the adults are often sessile on the substrate. The diversity of crustacea is quite baffling. You look at some things and it's very hard to imagine that they really are crustacea. An example of that are the barnacles. Barnacles are crustaceans, very closely related to the crabs, but especially adapted for being fixed to the substrate. These are crabs which are familiar to you. Again, they show a similar body plan. You've just got to realize that the dominant body part that you see is the cephalothorax. The abdomen in the crabs is small and is flexed underneath the cephalothorax. Here they have the limbs, some of them specialized. And the mouth, have a look here and you'll see these are mouth parts. They have a number of different mouth parts which are also modified limbs. So this is a fairly primitive condition. They're still showing, although they're more advanced than things like the trilobites, they're still the relatively primitive condition. Many of the crustacea are completely planktonic. They live in the water column. Those are all things like krill, which often form the basis of food networks in marine systems. All of the arthropods have to shed their skin. Now we know that because they are ecdysozoans. And the whole process of shedding skin is extremely complex and a very dangerous period for the organism. Very often they will go away somewhere, they will creep away and find somewhere to hide while they're shedding skin, because initially, once they've shed the old skin, as you see the spider has shed here, the new skin is the new exoskeleton is relatively soft and takes a while to harden. So the most advanced of the arthropod groups, and also one of the most successful in terrestrial systems, is the subphylum called the Hexapoda. Hexapoda simply means 6 legs. And these are the insects and all of their relatives. As they have there are more species of insect than of all other animal forms combined. All other animal forms combined. And they live in many different habitats. Most importantly, most importantly terrestrial, of course, but then also in fresh and in marine water as well. Excuse me. So where was I? Yes, so most importantly, in terrestrial habitats, we see this huge diversity, but there are many freshwater insects as well. And there are a few marine insects. It's not a habitat that they have become particularly important in, but there are a few marine insects. One of the most characteristic aspects of the insects is their ability to fly. And flying is always a very important characteristic to acquire, because it opens up so many different habitats for an organism. They can move from one place to another very easily, quite apart from the fact that they can escape predators, they can find food, and they can find mates more easily if they can fly. So the adoption of flight is really tremendously important, and one of the things which has contributed most significantly to the success of the insects. Now the insects, first of all, the thing we notice first of all about them is they still have the basic arthropod body plan, but they have reduced the limb number. That's where the name comes. Hexapoda means 6 legs. So they have reduced the limb number considerably. There are no more abdominal limbs and those sorts of things that we saw in the crustacea. The wings are a development of the thorax, and they are not limbs. They are not modified limbs. It's an entirely new adaptation in the insects. There are various kinds of wing in this group here, the Coleoptera or the beetles. The outer wing is a hard protective wing. The second wing is a membranous wing, which is folded underneath there when the insect is still and not flying. But in many of the insects, the wings are permanent. They're not, are this similar and permanently unfurled, not like the coleoptera. So this, by the way, is a dragonfly, one of the most important predatory insects. All right, so this those are the ecdysozoa. Now we come to our last group, which are the deuterostomes. Now this is our own group, and please look up the difference between the deuterostomes and the protostomes again, but I will just remind you in a second. The deuterostomes are a very diverse group because they include a number of the invertebrate groups as well as our own group, which are the chordates. So just to remind you that the deuterostomes we all show the same kinds of embryonic development. First of all, they have radial cleavage of the zygote. Second of all, the anus develops from the blastopore. In the protostomes, the mouth develops from the blastopore. The next really important thing is that the deuterostomes show similarities of DNA sequence, which link them together and distinguish them from all of the protostomes. Here's the deuterostome development. Here is the here's the protostome development. Here is the deuterostome development. Please make sure that you understand these and have them memorized. So the invertebrate groups in the deuterostomes are the echinoderms. And this is the one group. We're going to talk about another invertebrate group in our next class, but this is the last of the true invertebrates. These are the echinoderms. And these are marine groups. And there are a number of different groups, but they all have some important characteristics in common. First of all, most you'll see are built on a five-fold plan. 1, 2, 3, 4, 5. This is a sea urchin. 1, 2, 3, 4, 5. But it may be on multiples of 5. A pentamerous body plan. Second of all, they appear to be radially symmetrical. Now, this is rather strange. It's radial symmetry, which is completely unrelated to that of the Nidarians. It's actually a secondary radial symmetry, and it's not actually a true radial symmetry. Their larval forms are bilaterally symmetrical, and when we look at the carefully at their morphology, we find that they're actually not completely symmetrical. Although the overwhelming appearance is radially symmetrical, when we will find that there are some organ systems on one side that do not occur on the other. They are fundamentally bilaterally symmetrical, and they have adopted radial symmetry secondarily. It's a bit confusing. The next thing is they do definitely, most of them have a tough skeleton. And this skeleton is based on calcium salt. And looking at it, we might be tempted to think that it's an exoskeleton. It's not. It's an endoskeleton. This calcium carbonate and some other calcium salts as well that make up the skeleton in the echinoderms is actually an internal skeleton. There is a thin epidermis that covers the whole thing. Where is our skeleton? Our skeleton is also calcium. It's actually calcium phosphate in the case of the vertebrates, but it is an internal skeleton as well. So there's immediately a similarity that we need to think about. So there are a number of different groups. These are called the asteroidea, the sea stars, 1, 2, 3, 4, 5, again, pentamerous. I'll come to this one later. These are the sea urchins. These are called the feather stars or the crinoids, a very, very, very ancient group. They appear, we find them early on in the fossil record. So the same with the brittle stars here, which are called the Ophiuroidea. They show some other similarities. They have some other derived features amongst all of them. And the most important of these, quite apart from their pentamerous symmetry, is the fact that they use a special locomotory organ, which is unique to the echinoderms, called a tube foot. They have numerous tube feet, and you can just make them out here on the sea urchin here, sticking out. They are soft, tube-like extensions with a sucker on the end, and they can be fixed to the substrate and actually move the organism around. They can also be used for capturing and dealing with food items. Sea urchins, their tube feet, they will move items that land on the echinoderm. They'll move it down if it's suitable for being eaten. And then the tube feet on underneath actually move them around. Most of the sea stars, the asteroidea, are highly predatory, and they usually prey on bivalves, and they use their tube feet to clamp on either side of a mollusk and then prise it open so that they can eat it. This group here are called the sea cucumbers. Also, the name is Holothoroidea. And this may be a little bit difficult to link these tube-like organisms with everything else. These all seem to bear some strong similarity to one another, especially when you count the number of legs that they have. But these look more worm-like. They actually still have an internal skeleton. It's just that it's not rigid. They have plates, little tiny plates embedded in below the epidermis, but they also have tube feet that they use for locomotion. And if you cut them across, down cross section of them, you'd see that they also have a fivefold symmetry around here, the outside. So these are the major echinoderm groups, and these are our kissing cousins. They are in the same group as we are, the deuterostomes, and they show molecular similarity to all of the rest of the deuterostomes, and they show some of these characteristics, like having an internal skeleton based on a carbon compound. I mean, on a calcium compound, not a carbon compound. Okay, so the very last group in the deuterostomes that we're going to spend some time talking about in our next class are these, which are the chordates, and that is our group. There are still one or two invertebrates that we need to talk about, even in the chordates, but they are barely invertebrate. So we'll talk about that next time. I want to remind you all, please, that first of all, that assignments are going up once a week, and many of you are already falling far behind on your assignments. So you are pleased to make an effort to catch up on those. You have 2 weeks for grading, okay? The assignment is available for you to use for the rest of the semester for revision and everything else. But for grading, you need to complete within 2 weeks. And that is a very significant and important part of your overall grades. Second of all, there will be a quiz on Wednesday. We'll have a quiz on Wednesday on material that we have covered so far. And it can be anything from there. Remember that your first class test, I believe, is on the 14th of February. So this is an attempt to get you to start doing your revision already. And I will draw questions for the quiz from the assignments that have been set for you, okay? So I will see you then on, hopefully, if they've got the college dried out, I will hopefully see you on Wednesday. Okay.